Hi, today this video we see about proteomics using Expasy tools. What is proteomics? Proteomics means it's a large-scale study of proteins and the structure and physiological role or functions. So we will study the proteins by using Expasy tools. The term proteomics first appeared in 1997. So using this proteomics, we can able to investigate protein expression patterns at given time in response to a specific stimulus, but also to determine functional protein networks that exist at the level of the cell, tissue, or whole organisms. And after we study the rates of protein production, degradation, and steady-state appearance, how proteins are modified, that is, we can able to study post-transition modifications such as phosphorylation, etc. The movement of proteins between subcellular compartment also studied using this proteomics. The involvement of proteins in metabolic pathways. So we can able to see how the proteins will be involved, converted in the metabolic pathways. It also easily studied through this proteomics in our tools. How proteins interact with one another. That also we can able to easily study through, understand through the proteomics. By using this bioinformatics of studying proteins, the tools will easily used to identify the functions or any properties of the proteins, and all this investigations are being done through the proteomic tools. So let's see about what is Expasy. Expasy is an online bioinformatics resource operated by this SIT, Swiss Institute of Bioinformatics. It is an essential and integrative portal which provides access to over 160 databases and software tools and supports a range of life science and clinical research areas such as genomics, proteomics, and structural biology to evolution, biogeny, and systems biology and medical chemistry. The individual resources, that is databases, web-based, and downloadable software tools are hosted in a decentralized way by different groups of the SIP, Swiss Institute of Bioinformatics, and partner institutes. This was first created in August 1993. Originally, it was called Expasy, means Expert Protein Analysis System. So Expasy means it's an Expert Protein Analysis System and acted as a proteomic server to analyze protein sequences and structures and two-dimensional gel electrophoresis. So we can be able to analyze primary, secondary, tertiary structure all through Expasy tools. So common procedures for how to use this Expasy tool means go to the Expasy. It will lead to the Expasy homepage. We have to type what are the tools we need. Then when you have to search the tools and find the tools, and then you have to click the tool. It will take you to the homepage of the proteomic tools. Here they have one small box. Then you have to paste your protein sequence, interested protein sequence or accession number. Then you can click compute parameters. Sometimes in some tools they may be shows the perform. So you paste the sequence and you click perform. Or otherwise, sometimes you have to translate the sequence. The small box will be there. That is after pasting the sequence, you have to click translate. So this is the common format for procedure. So first we see about the physiochemical properties of protein sequence. So the tool used for studying analyzing the physiochemical properties of our protein sequence, we have to choose a tool as protoform tool. Proto is a protein parameter. It is shortly referred as a protoform tool. By using this tool, we can be able to compute the various physical chemical parameters for a given protein stored in SwissProt or TRMBL or for user-oriented protein sequence. That is, we can able to analyze or compute the parameters. The sequence maybe we can able to retrieve from the SwissProt or TRMBL or our own that is our synthesis the protein sequence. Also, we can able to evaluate it by using this protoform tool. So, in protoform tool, we can able to find out the molecular weight, theoretical isolated point, amino acid composition, atomic composition, extinction coefficient, estimated half-life, instability index, aliphatic index, and grand average of hydrophobicity. This all we can able to study in the or seen analyzed in the protoform tool. So it's for the procedure. We can go to Google search, type Expasy, and then enter click Expasy, go to Expasy page and enter specific proteomic tool. Or otherwise, we can go to the Google search and then type as the protoform. It will directly to the, when you click the protoform, it will get to the protoform tools homepage. So here we have to paste the sequence, our industrial protein sequence, and click the compute parameters. Then we get the results of all the parameters of our protein sequence. This is one of the proteomic tools. Second one is we can be able to find out the peptide mass fingerprint of protein sequence. That is, peptide mass fingerprinting is also known as protein fingerprinting. It is an analytical technique for protein identification in which the unknown protein of interest is first cleaved into smaller peptides whose absolute mass can be accurately measured with mass spectrometers such as Monditoph or ESR-TOF. So these are the way we were able to identify the peptide mass. So peptide mass means it cleaves the protein sequence. The protein sequence maybe we can retrieve from the SysProt or our user-entered protein sequence. Then we have to cleave this protein sequence with our chosen enzyme and compute the masses of the generated peptide. The tool also returns theoretical isolated point and mass values of the protein of interest. If desired, peptide mass can return the mass of peptides known to carry post-translation modifications. So this is also we can able to that is we can search through the peptide mass. Here also we have to paste the sequence and then we have to choose the enzyme, which type of enzyme can be used for cleaving this protein sequence. So enzyme means trypsin, chymotrypsin. So you already know each enzyme have its own cleavage site. So select this enzyme and then you have to perform. So from this we can be able to find out the how many peptides are cleaved and then which peptide having a high molecular weight or which peptide have a low molecular weight. This will be seen in the peptide mass fingerprint. Next we see about peptide cutter. It is also similar to that of the peptide mass fingerprinting. So it's the same one. We have to choose the protein enzymes. And another one tool is Peptra. So what is Peptra means? We have to put the just we have submit the protein sequence. It will draw the peptides. So we have to give a primary sequence. It will form a linkages with the protein that we given protein sequence. It form a structure, peptide structure image. That tool is Peptra. Then next one is Proteus. That is the combined transmembrane topology and single peptide predictor. So here also we have to paste the sequence and then we can be able to find out the transmembrane regions. Next one is translate tool. So in this tool we have to give a DNA sequence. It will translate it into the protein sequence or RNA sequence. It will that. Then we can be able to find out the by using this translation tool. We can form in the DNA sequence summation. It will contain the protein sequence. So from this we can find out the open reading frames or mostly in the red form. And then it will help you for the selecting for the initiator of the amino acid sequence. So this is about the translation tool. And next one is detection and alignment of repeats in protein sequence. So we have to find out the repeat of sequence repeats in the protein sequence can be studied by using the tool radar. You have to detect and alignment of repeats in protein sequence can be detected by using the tool radar. So for the radar sequence, we have to submit it. We can get the how many, what are the sequences having a repeated sequence, where it is the sequence will be repeated, will be seen in the result. Next is the secondary structure prediction. That is, we can use the SOFAMA, or another one is another one type of secondary structure prediction. That is GOR, GOR. It is also we are using a tool. Another one tool is for the secondary structure prediction is CFSP and Choseman House, and it is also used. So there are three different type of secondary structure tools will available. Third one, so secondary structures are the beta helix, alpha helix, the random coil. So in this sequence, we can be able to know what are the secondary structures may be present, and then which type of secondary structure is predominant in the given sequence that should be studied or analyzed. So other than that, secondary structure, you can able to see in transmembrane regions. So for this, we have to use the tool is predictor. TM predict, that is a transmembrane prediction. So we know the transmembrane, that is the protein will be present inside the lipid layer, another will be first layer, that is called the transmembrane regions. So these are the tools we have studied in this seen in the video. So next we go to the